So today we are fortunate to be joined by James Valentine and Richard Lewis. James Valentine has spent the last 17 years working in the regulatory space, including 6 years at FDA, where his role was to incorporate the patient and caregiver voice into benefit risk calculations at trial design and approval stages. And for the past 11 years, he's been a partner at Hyman, Phelps and McNamara, which is a boutique FDA regulatory law firm in Washington, D.C. Their firm acts as a sort of regulatory strategic partner and has experience in handling regulatory challenges for a variety of different types of sponsors, disease indications, and development stages. Our other speaker is Richard Lewis. He has many years of FDA experience as well. He worked in CMC for 5 years and was a facility inspector for CBER. So he brings that specific expertise to HPM and does a lot of the CMC manufacturing process development for their clients, helping them with their independent reviews of data and facilities prior to their FDA submissions. James has a few slides that he and Richard are going to go over. So if you would like to share those now, and then we can get started with questions. I will mention that this is an open Zoom call, so anybody is feel free to raise your hand and be called on at any time, and we will call on you and you can ask your questions. We will also be monitoring the chat if there are any questions in there as well. So take it away, James.
Sure. Well, thank you so much, Karen, for the nice introductions, and it's really a pleasure to be with this group today and have an opportunity to help answer some of your questions. Richard and I think you heard a little bit from our background. We're working with a wide range of different product types at different all different stages of development. And so a lot of our work involves some of the most challenging kind of development cases, which often are in rare diseases. So kind of as we're getting started here, the different insights that we're kind of bringing to you here, come from working with many different product developers, many who are either themselves academic sponsors, a lot of programs that came out of academia that are now have been partnered or taken over by commercial developers. And so we also bring a lot of our lessons learned from those specific experiences. I know some of your questions that we'll be getting to really are focused on that. But as we're thinking about some of these early regulatory considerations, we're asked to focus really on in the pre-IND setting. We wanted to start first with some kind of opening reflections. Richard, maybe you can kick us off here.
Yeah, sure. So good afternoon, everybody. Happy to be here, and hopefully we can give you some additional insight and get some of your questions answered. So I'm really going to be speaking predominantly from the CMC perspective. And for CMC, your goal is to ensure that the product you're making is safe and that it's maintaining, when you manufacture, the critical quality attributes such that it can be effective in clinic and help patients. So when you think of the universe of FDA's guidance and all of these meetings with FDA, there's a lot of information there. And in many cases, it's not as specific as we would like. It's because the answer to the big question that FDA asks is it safe and effective? And that allows you to get onto the market. It's actually a really complicated question that has a lot of variables kind of baked into it. So when you're thinking about a rare disease, gene therapy, FDAs can be thinking about the severity of the disease, the population you intend to treat, the potential benefit the population can get from your treatment, and the prevalence of the disease. So when we go through examples of guidance or other information or feedback you can get from FDA, you always have to keep those variables in the back of your mind, because even as you talk to other groups that are kind of in the same space as you, their experience is not necessarily going to be your experience. You always have to make sure that you're doing the correct activities in order to get to the next stage and not doing either too much or too little, because both of those will engender delays and a lot of additional costs. James?
Yeah, and that applies, I think, equally to non-clinical and clinical considerations. as you're, moving through the preclinical stage, towards the clinic, and then obviously through clinical development, context is key. The rarity of the condition, its seriousness and unmet medical need. It truly is important. It kind of might sound like, well, isn't every disease FDA sees, have a lot of these characteristics, but FDA does really take a nuanced view when considering those things. And so often it makes, every disease, every program so very unique and specific. And I couldn't agree more with what Richard said. It then kind of makes, the feedback you get from FDA feel like it's the exception to the rule that they describe in their guidance documents. That's, I think, in part why we wanted to start here, with just, kind of first at a very high level, reminding you or in case you weren't aware, mentioning, the types of interactions you can have with FDA in this pre-IND setting. I think pre-IND meetings are obviously the most well-known of these meeting types. But even before a pre-IND meeting, you can have product-specific engagement with with FDA in the form of the interact meeting. It's kind of easier rather than breaking out, what that stands for. Really just think of it as the pre-IND meeting. These meetings that aren't the pre-IND meeting that I'll be talking about are more narrow. So it's not as guaranteed that every program can have this type of engagement. But in the interact setting, this is when you've done preclinical work on proof of concept, but you aren't, haven't gone down the path of doing your tox studies, and your manufacturing product process is still being defined for clinical use of the product. But really what's key for interact meetings is having novel questions, things that go beyond the guidance. And so, that is very relevant then to these rare disease situations. And I know there's a great interest in gene editing within this group here, and so there is more likely to be novel questions that you'll be facing, given that this is a relatively novel therapeutic approach, and there aren't much in the way of precedents to follow. Obviously, pre-IND meetings are much more comprehensive, and is something that every one of all of your programs can benefit from. And they're intended to cover all of the different disciplines, different issues that Richard and I will be talking about today. But one thing you might not be as familiar with, that might relate to some of the topics we talk about, which really are things that are more in the category of what I would call enabling your clinical development down the road, are the development of drug development tools. So whether it's biomarkers, clinical outcome assessments, different trial designs, even conceptual approaches, FDA is willing to engage, in a non-product specific way to help move the ball forward with, some of those things. And these are, all different types of tools that you don't want to wait to develop until you're in the clinic or having those product specific conversations with FDA. And so, consider this and other, even more informal types of engagement with FDA to try to advance, some of these drug development tools earlier. And so, this is very high level intentionally because we might be referencing engaging with FDA as we get into your questions. But this is kind of the general framework for that. Another key thing we were asked to talk about is a little bit of just like, what are some resources? We're going to be answering some questions today, but where can you go, to get the deeper dive, and what's really kind of most useful or relevant? There's so much information out there. I mean, FDA's own website is immense and at times hard to navigate. So Richard and I pulled out what we think are some really key on this slide, key early phase resources. Probably the best place to start, especially for anyone that has not submitted an IND before, is that first bullet, investigational new drug applications, prepared and submitted by sponsor investigators. This is one of the few guidances that's really geared towards academic settings. And it's very comprehensive, in that it talked about all of the requirements across disciplines. and lays that out in a way, an approachable way, versus, expecting you to come with some level of expertise in every area. So even if you're really, you have a lot of expertise in, let's just say, the non-clinical development, but some of the stuff that Richard's talking about on CMC, is new to you, even before jumping into the CMC guidances, that sponsor investigator guidance will give you some grounding. And then I think, it's kind of the, from the non-clinical perspective, the elephant in the room is going to be your toxicology program. And so, FDA's, guidance on, toxicology studies, this is kind of the first guidance to start with, and will really lay out kind of FDA's expectations for the extent and scope of non-clinical safety studies. But I'll turn over to Richard to kind of mention, why he included these other resources that follow.
Yeah, the first one here I included, it's because it's, the foundational guidances that sets you up to answer the most fundamental questions about what you need to do from a CMC perspective for your IND. One, I guess, cute rule of thumb that I have is when you look at the titles of FDA's guidances, and when you start to see your actual product area in the title, it's a good indication you're going to get a higher, granularity of feedback from FDA. So when you read these other guidances, like the very foundational cGMP for phase 1, you have to keep in mind what I said just a minute or two ago, that it's not necessarily written with your patient population in mind, your prevalence, your severity, or the magnitude of benefit. So it's great to give you kind of the tools to start with. But the guidances are going to be most helpful are the ones that are geared more specifically to your product area. So the next one, actually, this is probably one of the nerdier things I'll say in my life, is one of my favorite guidances. So this CMC guidance, and it's for human gene therapy. It's a great guidance because it not only gives you some insight into the very scary electronic technical document framework, but it gives you a few sentences for each section what FDA expects to put in there. It's a great way to start building that checklist of what you need to do as you assemble your first big submission to FDA. And in addition to the guidance itself, I've put some links to other presentations and slide decks that give you better information. Now, potency is one of the key pieces of a biotechnological product. How you define potency, how you put specifications around it, because that really dictates whether your product can be used or not. So looking through that potency assurance guidance, getting familiar with it is a great thing to do in order to set up what are going to be your future questions in your first interaction with FDA. Potency should always be at the forefront of your mind really early on, and it's really not going to be resolved and settled until your NDA or BLA is approved. And I think we have one more slide on guidances.
Should be up now. Do you see it?
Okay, great. Yes, I do. So this one, we're not going to walk you through them, but it's one thing to illustrate that the universe of FDA guidances is very broad, very vast. And what we see as a pitfall for many sponsors is that they didn't know that guidance even existed, and searching for them isn't necessarily the easiest thing to do. So this is not, of course, an exhaustive list, but we pulled together other guidances for you to consider because they can just go unnoticed, and you may spend a lot of time, effort, and energy doing work you don't have to do, or asking questions you don't have to ask because the guidance already exists. And this touches upon a lot of the different discrete buckets for CMC and phase 1 studies.
And these slides, are available to be shared with you. And we did hyperlink to everything. We know some part of the challenges. Yeah, you could Google search every one of these, and that might be the next easiest way to find them. But everything we've included in here will be hyperlinked to. And then the last thing is, I think as part of this series that you have, we're happy to be, a resource, to you in the future. So when you get these slides, you can have our contact information. But what I'll call out here is on the bottom is our firm has a very active blog, multiple, if not daily, multiple a week, if not daily, blog posts on different issues. And because we do so much work, in the, broad, CBER and gene therapy and gene editing space, as well as in rare diseases, while not every blog post, might be of interest to you, there is a lot, especially emerging programs and topics that we post on. So would encourage you to subscribe to that as just another one of those resources. So with that, I'm going to stop sharing. We wanted to not spend time kind of, talking at you, but hearing your questions and, sharing our insights to that. So I'll turn it back over for, the Q&A discussion.
Yeah, thank you for that overview. We appreciate level setting. We have a couple of groups who've had a handful of interactions with the FDA, whether it was Interact or pre-IND, but we have other groups who haven't. So certainly helpful to have those resources available. So thank you for putting those together for us. We do have a list of some pre-submitted questions that we'll work off of, but certainly, just like Karen mentioned earlier, anybody who wants to comment on the discussion, we want to make this as interactive as possible, as much of a dialogue and conversation. Feel free to unmute, raise your hand, drop information in the chat as needed. So with that being said, our first question is, what are the traps and missteps that academic investigators and institutions in early development may make as they're preparing for a pre-IND? Any advice on how to avoid these?
Yeah, so I'll kick us off. So my comment again is going to be really focused primarily on CMC, and it is true. Being an academic institution, you can create your own delays or roadblocks as part of your general FDA process. And one of the more common areas that I found is by making assumptions about what FDA thinks about your program before you ask, and doing a lot of work based on that assumption, only to be told something later. And I'll give you a couple examples because that's not, that's hard to digest in the abstract. So, we've had academic institutions that, generate their own companion diagnostic for their clinical trial, and they do their own validation, and they, submit their IND to FDA, and they find out that what FDA is really looking for that disease and condition is something entirely different. So they have to go back to the drawing board, and from their perspective, entirely revalidate their assay, set different endpoints, and it can delay them by 6 or 9 months before they're really in a position to submit again. And something like that can be mitigated if you asked a very clear question, either at your pre-IND meeting or at some point earlier, about really what FDA wanted to see for that type of program. But the other side of the coin is also true. It's also that have to be flexible. So there have been a number of instances that prior to the IND, or really even the pre-IND, the institution does a huge amount of work to show that a specific impurity isn't at a level that's an area of concern, or they've done a lot of additional manufacturing and just process characterization, only to find out that FDA really wanted a fraction of that information for the stage that they were in. They collected 6 or 9 months of stability information when FDA would have been happy with 3. So it's important to keep an open mind, also keep an open mind on behalf of FDA, and make sure that you're not either delaying your pre-IND meeting because you want to collect more data, but also making sure that you are really kind of honest with yourself about what your pain points are, because those make the best questions for FDA. Now, and finally, and this is actually something that's a theme throughout a lot of things that we're going to talk about, is how to make sure that you maintain the continuity of your program. And what I mean by that is, you might have to manufacture one lot through your first phase trial. You might have to manufacture several. So as you do more manufacturing, as you make changes, you always have to ensure that you have a data bridge to everything that you do. So your non-clinical lots have to be, you have to create a data bridge to the lots you use in clinic, the lots that you use for your next phase, because we always have to make sure that everything's still comparable. So for an FDA's mind, that we're talking about the same product as you make different iterations and changes to what you're doing. So James, for clinical and non-clinical?
Yeah, no, that's, I mean, I feel like probably I'll say this a lot, so many things that Richard mentioned that, in terms of how you think about and approach FDA on CMC, apply across the board. But maybe one thing that's a little bit more specific, that I see as a kind of a misstep, and it kind of manifests in a lot of different ways, but it really comes down to, either poorly conducted or poorly documented, non-clinical studies. And really, I'm not talking in this case about tox. Pivotal tox studies are, usually done under GLP conditions, but more, pharmacology and proof of concept studies, which, are usually done earlier, often just, for, in some ways, for our own information and edification to see if we have something, in terms of the, our therapeutic approach that's worth, carrying forward, and whether it's an animal study or not, these, preclinical studies, have seen many times either be poorly kind of planned, executed, or documented. And I think that's because they were never intended to really inform regulatory decisions when they were done. But over time, more and more these types of, kind of preclinical pharmacology and proof of concept studies are being used more for FDA regulatory decisions. Early on, they're really valuable for making different types of benefit risk decisions, which can be really critical in terms of, kind of being the counterbalance to what we see on the toxicology side of things. But also, in terms of benefit risk, helping select what patient populations we're going to start with, and obviously what doses. It also, in a lot of rare diseases, gene editing is, well set up, and really lots of targeted, therapeutic approaches are well set up to have impacts on direct, disease, causative biomarkers. And, that creates great value, both, whether we're doing dose ranging and using them for, kind of our PK/PD assessment, but maybe even as, surrogate biomarkers, for an accelerated approval. And, in these rare disease settings, our first clinical trials, are expected to not just be classic phase 1 studies, and start to really contribute to evidence of effectiveness, but in order for FDA to be comfortable with use, for example, of different biomarkers, we often need to rely heavily then on non-clinical information to, support kind of their surrogacy, their predictive value. And so, so this isn't something that, we have time to maybe replicate higher quality studies, in parallel, to our phase 1 program, to kind of, set things up and establish endpoints for later stage development. And we really want to be, have that higher quality information early on. And so I think it's just a little bit of almost like a shift in how we think about even some of these really early experiments that you're doing, and start to say, well, this might actually have regulatory impact and implication. And so, try, I'm not saying every, pharmacology study needs to be under, full GLP conditions, but, start to think about how you, might want to, better define and pre-specify, your experiment, and, have more controls in place than you might otherwise have, because, we see at the time of the IND and then even beyond, FDA raising complaints about, the quality of these preclinical studies that, really could have maybe been, allowed a development program to proceed, years more quickly if we didn't need to now go and redesign an experiment and redo it later on.
So I guess to summarize all of that and all of this experience that you have, what would you say are, the number 1 or 2 absolute must-haves that you need to get that successful IND?
Uh, so I'll start for CMC. I mean, you know, if I'm to boil it down to one thing, I mean, you need to have a really well thought out and thoughtful pre-IND meeting, and you need to write, really clear, specific questions after doing a review of your process. And primary the paradigm is safety. So, what aspects of CMC are the most important for the safety of your product? There are times where there are specific standards or generally industry accepted levels for things that you can say, okay, I know I'm, within the right range for a certain attribute or not, but there's always going to be a few pieces that don't fit that mold. So writing clear, specific questions to get FDA feedback so you understand whether or not you need to make a tweak to your process, a tweak to how you do dosing. That for me is the best thing you can do to de-risk your first future IND submission.
I have the same same answer as Richard, which is really thoughtful and strategic FDA engagement. I think one of the things that I think about is I think for people who any sponsor and this is not certainly not only academic sponsors, many industry sponsors that don't have as much experience engaging with FDA have this idea that FDA is a monolith. There's a single FDA, and that couldn't be further from the truth. Richard and I both been at the agency. We know FDA is made up of lots of individual people, and so there's different organizational units. We have the big different centers, drugs and biologics. Maybe most relevant for our conversation. Within that, there's lots of different functional and teams, and even within disciplines, there's different divisions and branches. And ultimately, at the end of the day, even within a division or branch, you're getting assigned to a particular set of reviewers, and they all have different views and interpretations and takes on the same laws, regulations and guidances. And so, I think that makes that FDA engagement even more important, because even if you've had that experience in the past with a different program, a different product, one of the things that is maybe one of the most truisms about FDA is even people like Richard and me who are there on a weekly basis maybe with different programs, we can't even predict how FDA will answer a question. I mean, we probably are maybe the next pretty close to it. We get the flavor right, but there's so much inconsistency because of the nature of it being of humans being human and FDA being made up of lots of different people. And so, I think in terms of, the best thing you can do, number one thing you can do to have a successful IND, is to de-risk it through, that engagement with FDA. I mean, that's going to be not only valuable to you in having that successful IND submission, but having kind of confirmation of what you're doing and what your plans are, is gold. As you're maybe down the road thinking about, partnering or even just internally, getting buy-in within your institution to know that you're on the right path. I think those involved in drug development know about that inconsistency. And so, having, alignment on with what you've done and where you're going, is really, really valuable. Really it's gold. And so the other thing I would say that's maybe an extension of that, it's not the must have, but it's, maybe a little bit more aspirational, is to say, you're in a really unique position, in Richard, in my experience, as an academic sponsor, in reality, FDA kind of thinks about you a little differently, versus a, commercial entity. And so, I would encourage, and what that kind of means is, they're not going to lower the bar for you, but they tend to be a little bit more open-minded when they give feedback. And so I would kind of encourage you to, yourselves then be a little bit more open-minded. And when you go in and try to get more information, talk maybe a little bit further down the line of where you want to go and what you need to do to accomplish that. I think, drug company sponsors, the industry sponsors are going to always be very focused on, getting them through the next gate of development activity. I think you have a little bit more of an opportunity to kind of chart your path forward. So,
Steve, you want? Yeah,
Steve,
go
go
ahead,
ahead.
Steve.
Yeah.
Yeah, so, there's always this fine balance of trying to do the minimum that you possibly can to initiate a first in human phase 1 trial versus maybe call them shortcuts early on to do that then delay the program later on if you're trying to move to phase 3 or things, and it's always that balancing act. So, and sometimes the things you can do proactively at phase 1 to prepare yourself further on put a really large cost and burden, to doing the first in human. But are there any things that you can recommend that are maybe not as quite heavy of a lift that you could be doing, at that first in human stage that would set yourself up for success, or accelerated paths later on, like at phase 3, besides doing like a commercial drug
yeah, yeah.
manufacturer for your phase 1 or something like that, but are there reasonable steps that you can take early on?
Yeah, James, you don't mind? I'd like to go first.
Go for it.
If we're going to give you one thing that you can do and invest additional resources in early on to set yourself up for success long term, for me in the CMC world, that's your potency.
Yeah.
Potency is, and it might not be one aspect, it could be at least starting early on with a matrix of multiple ways to look at your product. Potency assays rise and fall throughout a development cycle. Some you might not be able to validate in the future, some might be cost prohibitive, but getting that established as robustly as possible early on can pay huge dividends, because when it fails, when you don't do it early enough, or you had a plan that you find out by the time it's to phase 3, it's not really a good potency matrix, going back and redoing that adds potentially years. So for CMC, it's absolutely potency.
Yeah, I guess if I'm, kind of, I think obviously there's a lot of things, but you're kind of saying what's the biggest bang for your buck? Buck being not only just money, but time and energy. That's a limited resource as well. To me, it's kind of, you want to do work to try to, and this is somewhat assuming you're in a space where there's not like a predefined, like very clear, here's our endpoints, here's what's meaningful on that, here's the right patient population. So much of the time, and even if there is in a small molecule approach, that might not hold true, based off of a gene therapy or a gene editing approach. I think spending time doing a little bit, and it doesn't have to be a huge amount of it's a scary term, the validation work for endpoints, whether that's going to be biomarker endpoints or clinical outcome assessments, whether that's assessing it, looking and seeing what's assessed in the clinic and doing, chart reviews or retrospective natural histories, or even better, if you got the ability to do, a prospective natural history study, but really, don't, I think, see so much that there's an assumption that what is used in the clinic for diagnosis, for kind of prognostic evaluation, is just going to be accepted by FDA as an endpoint in an interventional trial. And that used to be more true. But these days, now there's a, very sophisticated psychometric, approach to validating endpoints. And so being able to do some of that work so that way whether you have an endpoint and maybe it needs some modification, or maybe something that you or a colleague is developing, has the requisite legwork to where you can include it in phase 1. Because I think that's a problem, is when, it would be great if you're doing a phase 1, 2, 3. Certainly in some limited instances that has actually happened, but you don't want to be waiting to test your endpoints in your hypothesis testing studies, And you don't want to, it's really easy to get away in phase 1 and phase 2 with just throwing whatever endpoints in. FDA is not going to come back and say, no, we have issues with these endpoints, because that's not the study that's intended to actually test those endpoints, and they're usually just very permissive. And so it ends up surprising people, surprising sponsors at end of phase 2 when they're told, no, you kind of need to start from scratch with your endpoint plan. And so I think in the pre-IND phase, that is really an opportunity to do some of that work. That way you can, do some of that, testing of those endpoints in your phase 1 and obviously phase 2, but even in phase 1, you can start to do some of the psychometrics of seeing how understandable your, if it's a tool for your clinician or a tool for a caregiver, you will be miles ahead. I've seen programs be delayed years between phase 2 and phase 3, because of endpoints. And so I think when you're so early on, you're thinking about, like you were saying, Stephen, what's the minimum I need to do to actually get in the clinic? What's the top, minimum tox work? Pharmacology, almost doesn't matter in terms of like a true up or down of getting into the clinic. Richard mentioned CMC, but I think investing very early is gonna be pay off in terms of so much time and effort savings down the road.
Hi, this is Angie. I wanted to ask a follow-up question. So I think based on your experience working with academic institutions, for the ones that have successfully advanced their first in-human trial to later phase trial or commercial, what are the paradigm they have established? Have they had internal expertise in terms of their CMC and process development, or do you think it will be more effective if they have hire like an external partners in terms of consulting or outsource some of the let's say potency assay development? Like, what are the most effective ways you guys think that can make that happen?
Yeah, so let's start with potency first, then I'll answer general CMC. So I think the overwhelming majority of my experience, they farm out the potency to a lab that is well established doing potency assays for drug programs generally. You have to be, judicious in your choice, where you want to pick a lab that is in the same space that you're in. You don't want to go to a place that's primarily a mass spec lab and ask them to do, ddPCR. So you have to choose somebody that's done similar processes before, either specifically for gene therapy or the biotechnology products, because they're more accustomed handling the material, method development. While it is highly related, it is still different. And because of the frequency in that you'll use not only that development part, but that testing lab, it's typically long-term, or excuse me, in the short term, cost efficient to have it be that third party, because you use them and they need them after they establish a lab. Now, depending on how much you have to manufacture as you get into later stages, the volume of the testing you need to do, it can be more efficient to bring resources internal. But at the early stage, most often people will opt as an external resource. Now, generally for CMC, it's a similar but still different question. A lot of companies in the gene therapy space use either contract manufacturing organizations or contract development manufacturing organizations that are third parties. Again, the similar kind of thought process applies, that you want to pick the right partner in terms of you don't want them to gain experience in your niche of biotechnology on your dime. You want people that are experienced doing those unit operations. They'll have the necessary equipment and qualifications already on hand. You don't want them to have to reinvent the wheel specifically for your program. You want to be one of those kind of me too or me as well programs underneath the umbrella of what they do for other sponsors they have that are going through FDA, and there are a number of them out there. So it's important to do a lot of vetting, because delays can be engendered by picking the wrong partners in terms of the amount of the experience they have, or maybe for a manufacturer, their current compliance status. You don't want somebody that's been recently inspected and found to be grossly non-compliant. You don't want them to help you develop your CMC program because they have kind of their own problems themselves. In many cases, having that resource kind of ticks that box for CMC. Depending on the specific program, companies will then also opt to get kind of a third party that's not also the manufacturer, because there can be kind of points of disagreement or bringing in different areas of expertise. There may be instances where a contract manufacturer doesn't feel a lot's appropriate for a clinic, but you do. So having a second independent CMC voice can be helpful. So it's really fact specific. It's whether or not you need that additional help is really dependent on your situation, the manufacturer that you pick.
That makes sense. Thanks.
Along the same lines, we have a question about GMP material. So we all know it's often costly, especially for academic labs. So what specific parts of the IND package do you think is really well suited for GMP-like?
Yeah, so GMP or GMP-like is really in the eye of the beholder. People will call things GMP or not GMP, and I'm still scratching my head at times as how they've made that determination one way or the other. I mean, if something has appropriate review and quality approvals and their methods are validated, you could call that GMP. So it's important that you don't make things more expensive for yourself. And what I mean by that is you can purchase a product, it could even be your API in some cases, and it has about 45 different tests done to it to confirm, purity, potency, safety. It's important to make sure that what you're getting delivered to you is exactly what you need and no more. You don't necessarily need to have every lot of product tested for the presence of vanadium. That's where you can gain efficiency in GMP-like material. You always have to be critical about what you're supplying, and you always have to think about the question, what's its purpose, especially early. You, of course, want it to be well controlled, and you want to be safe, pure, and potent. But what does it mean to be safe? What are the things that we're concerned about potentially that's in this product that we really do need to test for? And in many cases, there are a lot of reagents you can purchase, and there are multiple iterations of it in companies' catalogs that have various degrees of testing. So it might sound great that this is their GMP version, but when you really look at the C of A, the difference between their GMP and what they're calling their non-GMP are tests that you don't even want or need because they're not actually addressing, safety of the population that you need. So it's important when thinking of GMP-like for let's talk about reagents in module 3, that you're not assuming that when it's called GMP, that's what you need and anything less is somehow not suitable. We've run into that on a number of occasions.
All right, moving on to another academia specific question, perhaps. And you might have already covered this with responses to Steve's question, but if there's anything else you want to add to this, just, going back to the limited funding, limited people, limited resources, if there's one or two, value add-ons that you would put on the academic side, is there anything else, outside what we've already spoken about, that you can, recommend to really invest more into in academia?
I guess I'll start. Okay,
Yeah, go ahead, Richard.
I'll start for CMC. And we've covered some of the topics already because we've talked about manufacture, we've talked about reagents. Potency assay actually also fits under this question here. It's something that's really good to put resources into early because it can help you not necessarily accelerate, but maintain the timelines you want at various stages because you don't have to go back to the drawing board and figure out a new way to assess your product. But another thing that's really important from the CMC's perspective actually is simply planning. And I'll explain what that means. So you'll have, at inception, a product that you've developed. that has, a specific API in a container closure. And as you go through your development process, invariably there will be changes. But it's really important to the extent that it's feasible to minimize changes as much as possible. So there could be solutions in the short term. Oh, we have this container closure the manufacturer uses, this seems fine for now. And we'll just change it later. But as I mentioned kind of a couple of times throughout our conversation, is that you always have to build a data bridge from where you are to where you're going and where you want to be. So whenever you make a decision that you have to change it later, you're actually then putting more work on yourself in the future because it's a new variable that you have to assess through something like a comparability protocol. So planning is really important because wherever possible, ideally you pick like your container closure and not change it really at all through commercialization, or you'll pick a certain set of reagents, certain pieces of equipment that you don't then have to force yourself to change later and have to a much larger comparability protocol in order to move it to the next manufacturer. Under that same kind of theme, in terms of planning, is retain samples. So this goes again to the topic of building a bridge to the next stage. So retained samples at any point in your program are worth probably more their weight in gold, because that's how you build that bridge to what you're doing next. So when you plan out the scale of what you want to manufacture, what you need for a clinic, you always have to keep in the back of your mind that you're building up or maintaining a sufficient inventory of what your current iteration of the product is to then go to the next stage. I have seen on a number of occasions in an academic setting where they'll come to us, or maybe they have a commercial partner interested, and they have great data. They've done a great job documenting everything. They have really well-validated assays for the stage that they're in, but they have 3 vials left. And they have really no ability to seamlessly build the bridge to what they want to do next. And that kind of forces you to redo things. So it's not that you have to invest for this topic a lot of time and money, but you do want to make sure that you are setting yourself up for success in the future by keeping this kind of reference material on hand and not exhausting it such that you have to go back and redo not just CMC work, but potentially non-clinical work and hopefully not any clinical work as well. James?
Yeah, so maybe thinking about things that we haven't talked about as much yet. I guess before even that, maybe one thing just to shout out is FDA's Orphan Drugs Grants Program. So if you're not familiar with this, FDA has grant programs. It's one of the really only few places where FDA is involved in kind of helping support research, and they have programs both for natural history studies as well as interventional studies. But obviously in a academic setting where you're resource constrained, every dollar counts. And I've even seen for small startup companies that it almost serves as like seed funding that helps them really get going, and it enables them to start clinical development. So FDA isn't thought of as much in terms of the funding, and I don't think gets a lot of applications relative to other funders like NIH. And so just a little shout out for that since financial constraints was mentioned. I guess one thing is, we were talked a little bit more aspirationally earlier questions about what more can you do that would be high bang for your buck. But for this question, I think maybe my answer is really make sure you're taking the most advantage of what it is that you are doing. So what do I mean by this? You're conducting certain laboratory research. Maybe you're investing in or funding development of certain non-clinical models and doing testing on those. I would say really don't skimp on documentation and record keeping for all of this, and I'm sure this exactly applies to CMC in that work as well. Because I think that ensures that even if you are somewhat limited in what activities you're able to do, and you have to prioritize, it ensures that when it gets to a time where that information is going to be needed, and it might not be until the NDA or BLA review, it might not be in the IND review that this really, is all of that important. But having, appropriate documentation and record keeping policies and, ensuring that you're doing that from like the earliest stages of your development work, is going to make, all of these pieces that are really critical, at different stages of development be able to be regulatory grade. And honestly, it's just something that we see, time and time again, when an inspection happens, record keeping, undermines, the ability of some important piece of information to be leveraged as part of a review. So it's maybe the most boring answer I'll give to a question all day, but, I cannot emphasize enough, kind of documentation and record keeping.
So I actually have a follow-up on that one. So can you comment about Part 11 compliance then in regards to documentation? Because that certainly is something, at least our institution is having a huge conversation about, but, what from once you get to that NDA or BLA submission space, how are those documents scrutinized from that level?
Yeah, I'll take this one. So later stages, it becomes really important. There have been a number of instances where we've had, FDA inspection, go through your records and realize there's a critical gap in that, data doesn't have, appropriate audit trails or controls or backups, that they're not able to independently verify to original source the data that you submitted in your application. And there are really only two good answers to it. It is to do a real forensic analysis to recreate the data from hopefully paper records that we would all hope still exist, and they're from notebooks that aren't that have been appropriately controlled and signed and documented, that still of course still exist themselves. But the other answer is that you have to regenerate the data, and that is a really big endeavor. So Part 11 compliance is extremely important in the late stage. FDA inspects you not just for your compliance with the Good Manufacturing Practices Regulation, but to verify the accuracy of the data in your submission. That's actually the other half of an inspection. So it's how you've done it is going to be really important. Now, if you're still in 2025 or 2026, an entirely paper-based system, it could be that it's the process of just verifying paper records, but companies and institutions now use electronic batch records and are going mostly electronic. So if you haven't done that properly, you really don't have a submission anymore.
Yeah, really, I mean, and just to add one small piece to what Richard said is, if you're not, able or based off of, the work that you're doing, able to, be in Part 11 compliance, even for, preliminary work, think about having SOPs in place that dictate, that record keeping of paper records, because that forensic analysis, there can't be assumptions about how things were kept or documented or changed in paper. Nothing ever is going to prove that there wasn't some kind of, tampering or issue with a paper record. But, the fact that, you were thoughtful and approached paper record keeping, in a systematic way, will go a long way in those situations where you are relying on that paper record trail. So, because at the end of the day, a big goal of this, is to be able to, understand, what is this, the true source data. And if there becomes a point where there's a question, and it requires going to paper records, you really, really want to make sure that there's not, some red flag that is reached by an inspector that suggests that, your paper records are not reliable. So, even before transitioning to electronic, ensuring that your paper process, is, has, proper procedures in place, will go a really long way. So just maybe a practical nugget on that side.
Yeah, just a quick additional comment before we go to Stephen with the raised hand. This is an aspect where if you're using paper records and you plan to transition to electronic, I think a solid recommendation is to be a hoarder. All of those pieces of paper should bring you joy, and you throw nothing away. You should keep them for, you really keep them until you don't need them anymore, which is usually something like a 10-year cycle. So just because you switch to electronic, keep those paper records, please keep them.
Well, so I had a different question, but I'll ask maybe a quick question first. What is the time that you should maintain specimens from your preclinical studies and, and sort of paper records? Like, when is it okay to throw those out?
So I will give you a partial answer to that question because it is spelled out somewhere. I just don't know the actual answer off the cuff. So it's usually after the study is complete, it's a certain amount of time. What I can give you that's slightly a satisfying answer, I think it's no less than 10 years, but there is an answer out there. I just can't tell you off the cuff.
Okay. So my real question was, in honor of Halloween tomorrow, do you have any anonymized horror stories that you can share that are maybe case examples that maybe could have been easily avoided?
Are we talking about for the
Regula-
purposes of
regula-
data integrity?
regulatory blunders, or you know, kind of approach to an IND, or, I don't know, just some kind of anecdotal, anonymized horror stories that can serve as like a tangible example to people.
We also love site inspection horror stories as well. So
God.
Okay, site inspection horror stories. I mean, the classic one is when you arrive at the facility and you realize that only a few people there, and they take you to the audit room and you pass. There's a room full of people just loading paper into trash bags. I mean, that's a classic site inspection horror story of obviously they didn't clean things up before you got there. It is never a good feeling when you're in a facility that should house 1,000 people and you can't find anyone, because then you have to question where they are. That has happened. And it's not that that's only happened once. It's actually very common that every FDA inspector has that story. So sadly, that happens, more than once in a blue moon or full moon if we're keeping with the Halloween theme. James, do you have any that comes top to mind?
Yeah, I mean, I'll just quickly just mention another one, because I was actually thinking this in my head as you were, making your comment about, keeping paper records should bring you joy. Don't throw anything out. Thinking of like on the clinical side, a BIMO inspection, during a NDA review where, the inspection was announced, and between that time and when the BIMO investigators showed up to the site, I think there was a panic that maybe SOPs weren't being followed, and there was a cleanup that happened, but the cleanup ended up involving deleting source documents. And you couldn't imagine a worse timing than thinking, oh, we must be deleting bad data because we knew investigators were coming. That's you can imagine what the FDA investigators thinking, like, well, this timing. And you on our side understood from talking to these people that they thought they were doing the right thing of trying to like tidy up, but in rushing, mistakes were made, and the timing couldn't have looked worse, in terms of making it look like there was some nefarious reason for doing that. But kind of outside of inspection related, horror stories, there's kind of like this whole category of like reason for clinical hold that people just never, companies, sponsors just never think of. We all think of, the toxicology and having an appropriate margin and, safety related clinical hold issues. Obviously, once you're in the clinic, safety issues popping up. But one that has really plagued and has happened more in CBER than in CEDAR are clinical holds on the basis of prospective direct benefit. So this is in situations where you're developing in pediatric conditions. Most, I think it's like 3/4 of all rare diseases are pediatric. So these are the kinds of things that you'll all be facing. When you're doing trials in kids, there's extra pediatric regulations around to protect children, given their inability to consent themselves. There needs to be a demonstrated prospect of direct benefit to them in order for them to take on that more than minimal risk that's associated with any investigational product. And so the classic way that, in small molecule drug development is you go and you give the small molecule to, adults and get some kind of pharmacodynamic or, even better if there's like symptom relief, some kind of clinical benefit. You come back and you tell FDA, hey, we've got this, now we can move into kids. When you're doing gene therapy or gene editing, you're not going to just put in adults for the sake of putting it in adults. And obviously, normally there's a really good reason that you want to go into really, probably the youngest kids or, or as early as you can in the disease progression to be able to really see a halt of disease progression. Obviously, those are for certain types of diseases, where that would be relevant. But man, kind of assuming, that FDA is just going to allow based off of that rationale, that, it makes sense. You've seen in other programs, early intervention, being what offers that benefit to patients, has led to clinical holds. Usually there's some amount of, non-clinical pharmacology data that's submitted, but it's not really thought about and set up and described in a way to, justify the prospect of direct benefit. And non-clinical data can be the basis of prospect of direct benefit. You don't have to go to adults first. That old paradigm, isn't something that has to be followed, but, those studies were never designed to try to prove or show prospect of direct benefit. And so that's kind of, one where, programs are put on hold, and then oftentimes they're trying to come up with, a non-clinical model, and there isn't one, or, there isn't one with a strong enough phenotype, because FDA really wants to see phenotypic impacts, not just pharmacologic ones, to conclude there's prospect of direct benefit. I mean, and those have been absolute, tomorrow being Halloween, absolute nightmares. Just because, there wasn't this connecting of the dots. This is pediatric, just because we're gene editing, just because we're gene therapy, and there's a really good rationale for going early, there still is an evidentiary bar. And that has unfortunately really, come out, and been way too spooky.
Yeah, I'm going to add in the same vein of other topics for clinical hold that we haven't addressed. And it's still technically CMC, but it's actually a device. So the reason why I'm bringing up devices in the moment is that we've been focusing on like a gene therapy product that you're making. You're going to get to the point where you have gene therapy in a vial, but that vial, the therapy needs to get out of the vial and to the patient. And a lot of programs forget about that last bit of the journey from vial to patient. And that shows up at times in clinical holds. So how is your treatment getting into the patient? Is it going to be a simple intravenous injection? Is it something that's slowly infused into the brain? Depending on what the answer is, there's going to be an expectation from FDA that you assess that portion of it as part of your IND. Is it a pump? Have you validated the pump? Have you ensured that the pump doesn't go above a certain pressure let's say it's intracranial infusion. There's a tube that you've used. Have you done extractable, leachable testing on that tube to show that, for the compartment that you're infusing, you're not going to introduce any sort of, plastic or other impurity into the patient's brain? So that's an area that gets missed actually quite frequently. And, when I review IND clinical holds for CMC, it's probably the third or half the time there's a comment about the device in there, how it gets into the patient. So it's really important to keep in mind that you identify and establish clearly how you're getting it in, whether it's an infusion set, pump, and that if there's any data that you need to generate that you can't get from the manufacturer, and then manufacturers do this extractable and leachable testing, it has been the subject of a number of IND holds. And that's spooky, to keep it the thing.
So I wanted to take this opportunity to ask a question that was brought up to us by one of our coordinating committee members. There is a new process on rare disease evidence principles and a new plausible mechanism pathway to be published in the New England Journal in about 2 weeks. There's also previous guidance related to IND submissions for individualized ASOs. Would the panelists speculate on the future of rare disease or individualized genome medicines based on these new developments?
Yeah, I'm happy to kick things off here. So, for those not familiar, the rare disease evidence principles, it's kind of an awkward name, but the idea is to kind of at least propose a pathway for developing drugs for very targeted drugs for very specific types of diseases that are very rare. Less than 1,000 people in the United States. The new plausible mechanism pathway we know less about, but when the commissioner has talked about it in the past, this has been really talking about almost like individualized or very small handfuls or very, very low prevalence conditions where maybe in those situations some information about the impact on the mechanism or the pathophysiology with a well-established kind of mechanistic information plausible in the clinic but maybe established non-clinically might be enough to actually allow a product to the market. It almost sounded to me like the animal rule where you would in a disease where, or not a disease, but a scenario where you have bioterrorism or some kind of natural outbreak that is not naturally occurring at this moment in time. It's unethical to submit people to those conditions in order to test, and so you can evaluate using non-clinical models and then essentially do field tests, a registry of people who are actually treated once the product's approved. That's what it sounded like the new plausible mechanism pathway would be, but I guess we'll see when that New England Journal article comes out what it really is. This is all exciting. Obviously the individualized ASO guidances and all of these things are trying to say, how do we actually navigate development? We're dealing with very small numbers. Rare diseases is not one size fits all. When you're dealing with something that's 150,000 patients in the US, that's extremely different than dealing with 100 patients in the US. In my mind, I think of it really as like there's this big sliding scale. What these types of initiatives are really saying, it's not necessarily saying there isn't a sliding scale, but we really have to think differently, and maybe it's easiest to think differently first about those most rare situations, whether it's 1,000, 100, or truly individualized. And my hope is that our lessons learned from these types of initiatives will then translate to us figuring out, well, how do we then slide the scale the other direction? Because classic drug development is everything is designed for prevalent conditions, and we're trying to slide the scale downward. So I'm very excited about these developments. I think it'll obviously all their success will be relative to how thoughtful the processes that are put in place really are. But I think we shouldn't have to do a classic tox program, for a situation where, individuals, have very serious or life-threatening diseases, and there's, so few individuals that, we're not talking about exposing large numbers of people. And is there a place where, by requiring that, the cost benefit is so prohibitive that we're actually not, developing those products. The same goes for everything else. I mean, I talked earlier about validating endpoints. Like, what does that even mean in a situation when you're, only developing a product, in a handful of people? Does it even make sense to randomize? Randomize isn't curing any baseline imbalances when you have such a small sample. And, isn't that a small enough number where we can really just look at every individual person and ask the clinicians at FDA to do an in-depth review of what their experience was? So you can tell, I mean, this is something that I'm very passionate about. I think, I'm hopeful that, this will lead to some trial and error, because I don't think that there's answers to all of these questions. And, and these programs and guidances, give us a opportunity to raise and propose things to FDA that normally I would say wouldn't pass the red face test. So I think it is allowing innovation to happen. It'll be incremental, it'll be trial and error, but I hope there will also be that moment in time where we pause, or multiple moments in time where we pause and try to see, what's working, what are the best approaches, and hopefully also then how do we apply some of those learnings to situations where we have 5,000 people? Because I think that a condition with 5,000 people, experiences many of the same challenges.
Richard, anything to add to that?
Yeah, so I mean, the takeaways and the hope that James was talking about for clinical trials, also you see that in manufacturing, and especially you have device components to your program. There are many rare diseases where FDA is asking for validation of a special collection device and an assay that uses more tests than would ever be run in the next 50 years, because there's only 200 patients in that population. The number of vials that you're testing for the various impurities and attributes use up 30, 40, 50% of the lot, and we're making it much more expensive and time consuming to develop these programs. And there are been instances where I've been really encouraged that FDA is taking a more of a paradigm shift, focusing on what's the best path forward for a rare disease, but it's not uniform across all programs in all instances. And if they were to take additional time to do that same type of effort for CMC, it would probably dramatically lower the cost and time to actually getting a product on the market for a lot of these programs. So every new program, every new acronym, all of that, I think is very encouraging, because it all contributes to that same effort of making something more appropriate for the rare disease space.
All right, so we've got just under 15 minutes left. Maybe if we can round out our discussion, thinking ahead to potential future partnerships, from the academic side, potentially transitioning, if things go well, go as planned, over to future commercial partnering. So how I guess I'll combine these two questions together. How might the academic investigators, how might we de-risk our programs for future commercial development? And in that description, are there some big hurdles or issues that you have dealt with in your past when helping programs transition from an academic setting over to the commercial setting?
Yeah, I'll go first. So for those two bits, I mean, we touched on some of the topics already. When you are going to have a commercial partnership, your data needs to be more than enticing. It has to be useful. For your data to be useful, it has to, of course, be appropriately documented very thoroughly. And then you have to be able to build that data bridge to whatever that commercial partner is going to transition it to. And in many cases for CMC, that's a new facility, a new manufacturing line. Now, the other piece of your question, which is the difficulty, is I've seen in many cases they'll transition it to a new facility, a new process, a new scale, and they're changing a lot of variables such that they've created their own higher hurdle in terms of showing comparability to what was done by the academic sponsor and academic institution prior. So there are many commercial partners that think in a commercial way, which is instead of a 20-liter batch that was run for your first phase trial, they're going to do a 500-liter. So it's striking the right balance for on the sponsor investigator side, making sure that you have a program that's not just exciting, but it helps them accelerate through to the later phases, phase 2 and phase 3. And then on the commercial side is making sure that you're not making the job harder by trying to, instead of doing it in maybe 2 or 3 stages, go from the really small academic scale to the larger commercial scale. That's the biggest thing for me, because sometimes it's not just difficult, but impossible, because with all the variables that change, the scope of that study is probably more broad than your entire manufacturing history or your ability to supply the appropriate number of retains and samples in order to build that data bridge.
So it's a good question. And I mean, I think maybe some of the things that we've spoken to before will de-risk programs, but in terms of specifically de-risking, with in mind this idea of making, planning for a future transition or partnership, and what de-risk there. And, I mean, I'm talking to the CEOs of, all different size, biotech and pharma companies, on a daily basis. And, the thing that has, it's been now years into my career, but one of the first things that really struck me about doing regulatory work was how important these regulatory interactions are to the business, and making investments. And so, I think maybe, one of the best ways to de-risk your programs, we talked about engaging with FDA to de-risk them, in terms of actually knowing that you're on the right track, you're doing the right things. But I would say, be very strategic and thoughtful about those FDA interactions with this other stakeholder in mind. Think about the fact that one day in some due diligence of your work, you're going to have people from a potential partner reading that regulatory correspondence and saying, did they get the right buy-in, and was there enough interaction? Part of it isn't, just reading what's on the paper, but, a lot of early phase programs say, oh, well, this is all, no-brainer. Like, we know what we're doing, and they don't engage enough with FDA. And that's really scary to partners because, especially in these uncharted areas, rare disease, gene editing, I don't think you can feel so confident that things are, tried and true. And we talked about FDA is not FDA, they're not a big a single person, a single entity. Well, FDA changes over time. And I mean, it's probably been the bane of Richard and my existence, since January of this year of, this, FDA today, every week to week, it shifts in terms of what they are and who they are. And, the regulatory philosophy being applied, and with loss of people and turnover of people, when I say FDA is made up of people, well, advice that was given by other people, those people are no longer there. What are the new people, or the people who were there but maybe were lower level, now left to make these decisions on their own without that middle management? So kind of reconfirming and having interactions over time, versus having really big gaps, will be very de-risking to, these, commercial entities, and along the same lines, think about, asking the questions that you've been asked by those partners in the past, so the next time around, you won't just be answering that question, but you'll be able to point to FDA meeting minutes that gave the answer to that question. And then I think it's also the opportunity. It's not just the de-risk, but I think I made a passing comment maybe earlier about you all as academic sponsors. I think have a lot of opportunity to talk about the future in a way that FDA would normally gatekeep and say, it's too premature to talk about this. I think you actually have a unique opportunity to talk about the future. Talk about maybe what the different stages of product development will look like in the clinic. Talk about what a future change to a CMO manufacturer would mean and timing of that as relative to your development program. Normally again FDA would restrict the answering questions that are more right in front of them. I think you all as academic sponsors can get a little bit more out of FDA. And I can tell you, if you're talking then to a company, a potential partner, they're going to be blown away by seeing more forward-looking input from the agency.
Yeah. So something you said, James, sparked with something in me that I was grasping at what I wanted to say, and you articulated beautifully. It is perfectly okay to answer a question to FDA where you get an expected answer of yes, and you knew the answer was going to be yes. Why that's important for CMC, a classic example is you provide a comparability protocol that is really well written. It's fantastic. But you ask FDA, can FDA comment on our comparability protocol for the transition to a new manufacturing site? And you can get meeting minutes that say, FDA, this plan appears reasonable, or FDA agrees that this plan is appropriate for a future transition to a manufacturing site. That looks wonderful for a company that wants to take your program out of the small scale, potentially clinical manufacturing site they're using to a commercial site, because they don't have to wonder, well, is this really going to work? Is FDA going to have issues with this? And you can tell a company, oh, we're confident it's fine, but you've already asked the question to FDA and gotten that feedback, their stamp of approval. That looks great to a company because it costs so much money to transfer a product. And if they had the confidence that if they were to execute per that plan, provided everything goes well, of course, that FDA would be happy with that data package, that means the world. So there are times where you know the answer, you're confident in it, but you can ask a question to FDA where you get an answer that you expect.
I think there is some balance though, right? A lot of the meeting types you are only able to ask so many questions and no more.
Mhm.
And do you want to waste a valuable question in an answer that is going to be yes. And I think that's a good example of like, here's an instance in which you may want to do that. I don't know if there's any others that come to mind in the last few minutes that we have, where you're like, hey, you only have so many questions. Here's some that like are a really good idea to ask, even if you kind of know what the answer might be.
I mean, for CMC, the big two in terms of risk of the program and costs. I mean, manufacturing is one, but for me, to really get in writing, they've agreed to your potency matrix. You could have had previous discussions about it, and maybe they've said, okay, we may have little concerns. You might want to elevate this one to your potency matrix, and that's in a previous interaction from a year or two ago. That would be one of the ones where I think it'd be worthwhile, to circle back with FDA. Our potency matrix now consists of these two assays. Can FDA comment on that matrix to support phase 3 or commercialization? That can be a worthwhile question to ask, even if you've already touched upon it before, getting a definitive yes.
Yeah, I mean, I guess maybe on the first point of you get a certain limited number of questions, but my experience is that most of the time, in meetings, not all the questions are being asked that could be asked. You're not actually like, capping out on, the true threshold of questions. And so I think part of that is, this instinct to not want to, feel like you're bugging FDA or distract them from maybe the things that are really important to you to get answers to, the less expected answer, type questions. So I think what you're hearing from Richard and me is, it's okay to ask more. One of the great things is you're going to get preliminary comments, and FDA is going to answer your questions, and you only need to spend your meeting time talking about the topics you want to talk about. So you get a bunch of, maybe more routine answers. You put those aside and you focus on the real meaty issues for the meeting itself. I mean, one thing that just kind of came to mind when you asked the question, is a lot of the times, with gene editing or even other gene therapy approaches, there's on the toxicology side, there's expected limitations in, taking these types of products into different animal species. And I think that, a lot of the times then those limitations are brought up once we have the results of those studies. To the degree you're actually engaging with FDA before you run your tox program, whether that's interact or a slightly earlier, maybe midstream in doing tox work at a pre-IND meeting, even if you don't ask the specific question about the limitation, when you're talking about and proposing your tox program, go on the record and raising those. It's the same thing that applies when you're talking about issues that come out of clinical trials and looking at, there's, odd things that happen in the data. If we thought that some of these issues might happen and we pre-specify sensitivity analyses, put it on the record that these are things that we think could happen, it's not like we're in this situation of having a post hoc, a fully post hoc discussion around it. But that happens a lot with non-clinical data, and FDA is just as suspicious of, raising issues and limitations and saying, oh, it's well documented, in the literature that this is a known issue. FDA is much less likely to, agree, that this isn't something that needs a specific issue that needs to be further evaluated for that program if you didn't raise it ahead of time.
Well, it looks like we're at 3:30. We, of course, will share the slides that our speakers provided to us. So, and that has their contact information. So any other burning questions that you didn't get a chance to ask today, our speakers have, said they're happy for you to reach out. But thank you, James and Richard, so much for sharing your expertise with us. This has been a really good discussion. And yeah, just thanks for being here, and this was really, really informative and helpful.
Well, thank you for having us, and I love the questions and a lot of the ones that were raised, by the, everyone in attendance. And I guess staying on theme, hope everyone has a great Halloween tomorrow, either gives out good candy or comes home with good candy with their kids. So.